This tutorial is going to focus on mutations, and mutations really are very, very important in talking about evolution and how it works, and the notion of variation in populations. In this particular tutorial we'll just take a quick look at what mutations are in general, just a couple of different kinds in general, and then we'll focus on point mutations, and then in a separate tutorial we'll focus on chromosomal mutations. Generally there are two kinds of mutations. There are spontaneous mutations, so I'll just jot this down, so spontaneous mutations, and a spontaneous mutation is a change in the genetic material that does happen, as the word suggests, spontaneously, that it doesn't seem to be directly caused by any external influence. So it may be just in the copying of the DNA during DNA replication that there is a mistake made and a wrong base ends up in the wrong place, or part of a chromosome ends up broken off or changed in some way, but it happens spontaneously. The second kind of mutations are referred to as induced mutations, and to induce something means to cause it to happen. So this is where some factor causes some kind of genetic mistake, either in a particular gene or in a chromosome structure itself. Common induced mutations are caused by things like radiation, so UV radiation, ultraviolet radiation, X-rays, gamma radiation from radioactive materials, and so on, can all lead to mutations or alterations in the genetic material. Various chemicals can be described as mutagens. So a mutagen is a substance that can cause mutations. So chemicals can lead to mutations. Let's have a look at the two basic different kinds of mutations. So first of all, there's what's called a gene mutation. So gene mutation is a permanent change in the DNA sequence. So is a permanent change in the DNA sequence that makes up a gene. These mutations can either affect a single DNA base, so they can affect a single DNA base, we'll look at those shortly, or they may affect a large segment of a chromosome. Either way, you have some change in that gene sequence that affects the functioning of the gene. Mutations are mostly always harmful. So they're mostly harmful because they disrupt the code on a gene, and therefore this almost always leads to some non-functioning protein, which in turn affects various chemical reactions or structures, and therefore this is almost always what they refer to as deleterious, meaning it leads to the deletion of that organism or that cell or that function. So it's important to understand that most mutations are harmful, but rarely, and in some occasions, mutations may be beneficial. So by sheer accident, a mutation may lead to a gene or a base sequence that actually produces a functioning protein that in some way leads to a useful phenotype, and this can lead to something new appearing in a population. This happens pretty rarely, that by accident something good actually comes of a change to a long-established genetic code. Sometimes mutations are referred to as neutral mutations, and neutral mutation is where a change occurs in the base sequence, but there is neither harmful nor benefit, neither harm nor benefit come out of that change. And so the mutation just sits in the DNA. This is usually the case in non-coding regions of the DNA, where it doesn't affect the production of functional proteins. So a mutation will lead to a permanent change in the DNA sequence, but it doesn't affect anything. So it's referred to as neutral. It's neither beneficial nor harmful. So these are a few important terms, but it's most important to remember that most mutations are harmful, but the ones that are beneficial often feed into evolution as a new phenotype. I think we'll leave this particular tutorial at this point, just the basic background to mutations, and our next tutorial will focus on base mutations, or single base mutations.